In today's lecture, so we are going to discuss about intercellular junction and communication. In previous lectures, I have discussed about the cytoskeleton structure. So some of the concepts from previous lectures we are going to use in these lectures too. So let me start with the basic concept. In cell junction or in intercellular communications, what are the most important things that we should know? So here in these lectures, we will see what are the basic features of extracellular matrix, what are the different types of protein present in extracellular matrix, what is cell junction, what do you mean by cell adhesion molecule, what are the different types of cell adhesion molecule that played an important role in the formation of different types of cell junction, and finally we will see what is the physiological importance of different types of cell junction. Let's start with the basic concept. What is extracellular matrix? Extracellular matrix is the non-living materials that is found outside the cells in tissues. If you see the cell structure, it has a different component. So, what are the different components of cell structure? Cytoplasmic area, cell membrane, outside cell surface membrane. At the same time, there are different types of organelles present in the cell cytoplasm. Nucleus is also present in the cell center. Along with this, we have seen there is special structures called cytoskeletons also present in cell cytoplasm. So here it is all about the cell matrix or extracellular matrix. So extracellular matrix is basically the part which is present outside the cell or outside the cell membrane structure, right? And the most important functional features of that cell matrix, it acts as a support system, right? If you see the structure of a tissue, there are numbers of cells that holds together, right? And the joining of all those different types of cells in a single tissue are carried out by or helped by that cell extracellular matrix. If you see the extracellular matrix structure, there are various types of molecules that are present in extracellular matrix. Some are called protein molecules. Some molecules are carbohydrate that attach to the protein, and some are adhesive molecules, adhesive protein. Now we will see what are the different types of protein present in extracellular matrix structures, and what are the proteoglycans, and what are the adhesive protein. You can see some structural proteins, mainly collagen and elastin are present as a structural protein in cell matrix structure. There are numbers of polysaccharides that attach to the protein and they form a proteoglycans. We will see how this proteoglycans is present in cell matrix. Along with this, adhesive glycoproteins. So adhesive glycoproteins is very vital because it allows the attachments of adjacent cell, right, in a tissue. So what are the adhesive glycoproteins? These are fibronectins, laminins, and integrins, right? Now see, this is what is the general organizations of the molecules in cell matrix structures. This thick line is nothing, this is the collagen, which is structural protein. Along with this is an elastin. This is also a structural protein, right? Other than this, you can see some branches. These branches are nothing. These are made up of proteins and polysaccharide called glycoprotein, right? Along with this, we can see some transmembrane protein and some thin fibers which is attached to the others. So these transmembrane proteins are called integrin, which is an adhesive protein, and that integrin is bound with the laminin along with the fibronectins. So this complex fibronectin, laminin and transmembrane protein, these are adhesive protein. If you look into the transmembrane protein structures, it is attached to the cytoplasmic molecule. Here cytoplasmic molecule says cytoskeleton, mainly filament structures, microfilament, either it is actin filaments or other filaments like intermediate filament, right? So it means in the cell matrix structures, there are numbers of molecules present. All those molecules are equally important for the formation of cell junction. We will see how this matrix involves to the formation of junction. See here, what are the most important functional features of this extracellular matrix? It provides the stability to the tissue, right? Which is very vital for proper function of any type of tissue or for any type of cell. It allows the communications. So for the transport of different types of materials from cell to cell, communications is very vital. So the cell matrix structures allows the communications process between cells. And what is the role of adhesive protein? Fibronectin and integrins are involved in signal sending and signal receiving. So sending any type of chemical signal and receiving the same is very vital for proper function of cells. So here we have seen there are some adhesive protein that played an important role. Now, moves to the main point, cell junction. It is nothing. It is the connection between cell, and that connections occurs in a specific site of the cell membrane, and that connection occurs between cell. That connection occurs between cell and cell matrix, right? So for the connection between cell and cell matrix, there are numbers of molecules called cell adhesion molecules is very vital. Now we will see what are the different types of cell adhesion molecule, right? And how the cell adhesion molecules allows the connection between cell and cell matrix. Cell adhesion molecules are the key proteins in this cell junction. On the outside of the cell adhesion molecules binds to the neighboring cell or the extracellular matrix. And on the inside of the cell, they links to the cytoskeleton via some other proteins called adapter. So you can see two different types of epithelial cell, adjacent epithelial cell. And between these two adjacent epithelia, there are numbers of junction. These are called cell junction, right? That keeps both the cells together. But for the holding of both the cells together, these cell adhesion molecules played an important role. Not only this, if you see, these junctions are attached to some important protein or filament that is present inside the cell. These are nothing. These are either actin filaments or the intermediate filament, which is a structural component of cytoskeleton. So it means the cell adhesion molecule, which played an important role in the formation of cell junctions, right? And in these cell junctions, these cell adhesion molecules or adjacent cell adhesion molecules bind with each other at the same time. And cell adhesion molecules also links to the some molecules present in the inside of the cell like cytoskeleton structure. So these are different types of cell junction, right? And these are different types of proteins. I will discuss in detail. So like tight junction, Adherens junction, Desmosome and Gap junction. See, what are the different types of cell adhesion molecules which played an important role, right? And where actually they are found. There are mainly 4 different groups of cell adhesion molecules. So these are Cadherin, Integrin, Selectin and immunoglobulin superfamily. If you see the structural as well as the functional features of all those 4 different type of chem, the features is little bit different from one another, but there the goal is same, the formation of junction between cell and matrix, right? Say cadherin is an important cell adhesion molecule which makes the connection between cell only, and it forms a very, very tight junction between cell, right? And this type of molecules are mainly present in epithelial cell, right? Mainly skin epithelia and intestinal epithelia. Integrins, another important molecule, cell adhesion molecule, which basically involves the formation of junctions between cell and matrix, and it is found in muscle and skin cell. Whereas selectin, it also forms the connection between cell, but the strength of the connection between cells here is little less or loose connection, right? So these type of connections basically found in white blood cells and blood vessels, or these type of cell adhesion molecules are found in white blood cell as well as the cells present in blood vessels. Other than this, another important type is called immunoglobulin superfamily. So it is basically involved with the connections between nerve cell and immune cell, right? So here it means there are mainly 4 broad categories of cell adhesion molecules played an important role in the formation of different type of cell junction. Now see, what is the exact structural features of these different type of cell adhesion molecule, and what are the other various characteristics? This is the first one called cadherin or E-cadherin. The cadherin presence in endothelial cell, the cadherin presence in epithelial cell, called E-cadherin. So if you see the E-cadherin NHS, this is the cell membrane of the first cell, this is cell membranes of the second cell. So as I mentioned, the cadherins is basically involved with the connection between 2 cells. So here the membranes of the first cell, here the membranes of the second cell, right? So at the same time, you can see the structural features of both the cadherin presence in 2 different cell membrane or cell membranes of adjacent cell are the same. It means it forms the homophilic interaction. So homophilic interactions or homophilic cadherins are those which forms the connections between adjacent epithelial cells where the transmembrane proteins are same. Here the transmembrane proteins is cadherin presence in both the membrane. And another important features of this cadherin is it is calcium dependent transmembrane protein. It means the function or the potentials of this cadherin proteins is influenced by the availability of the calcium ion in cell. The second one is called integrin. The spatial structural features of this integrin is it is a heterodimer, right? It is made up of 2 strands. One is alpha strands of integrin and beta strands of integrin, right? So here it is also a transmembrane proteins. The most important thing is it connects the cell with the cell matrix. This is the extracellular matrix region, right? So in the extracellular matrix area, as I mentioned, there are certain types of adhesion molecules, adhesion protein or adhesive protein. So here adhesive proteins is this one, right? And this is fibronectin. It means this integrin molecules, which is a heterodimer, it is attached to the protein, adhesive protein present in the cell matrix, right? Other than this, the third one is called the selectin or P-selectin. There are different families of selectin presence. I am just discussing about the broad categories that is called selectin. So the type of selectin are basically categorized in which cell types they are present, right? So as I mentioned, it is basically a mix, a loose connection between cell. And another important feature of the selectin is it always binds with some a sugar moieties. So this is a glycoproteins presence in the cell membranes of the adjacent cell, right? This is also a transmembrane proteins which acts based on the availability of calcium ion. I mean it is calcium dependent cell adhesion molecule. and it is a heterophilic. Heterophilic here is because the protein presence in adjacent cell membranes are not same, right? It is two different types of protein, right? So it means it is a heterophilic in nature. This one, the fourth one, is called immunoglobulin superfamily. If you see the structures of immunoglobulin superfamily, it is very similar to the antibody structure. That is why it is mentioned here immunoglobulin in nature. Immunoglobulin superfamily. So what is the most important features? Most important functional features? It is calcium independent cell adhesion molecule, right? Now, in brief, we can say Cadherin makes a contact between cell, Integrin makes a contact between cell and matrix, Selectin makes a temporary sticks, and immunoglobulin superfamily involves with the formation of a link between nerve cell and immunocell. Now, as I mentioned, cell adhesion molecules are mainly involved with the formations of different type of cell junctions. Now, question is, what are the different type of cell junctions? Yes, cell junctions can be classified based on their structures, based on their functions or localizations, right? But in a broad way, we can classify the cell junctions like tight junctions, Adherens junctions, Desmosomes, and Gap junctions. All these four are mainly involved with the connection between cells, right? Tight, Adherens, Desmosome, and Gap junctions. But hemidesmosome and focal adhesion, these are two different types of cell junctions which forms the cell junctions between cell and cell matrix or extracellular matrix. Clear? Now, if you see the classifications based on their functions, these are mainly occluding junctions, adherent junctions, communicating junctions. So, occluding junctions involves the tight junctions. I will explain in detail. Adherent junctions includes adherent junctions, desmosomes, hemidesmosomes. Whereas communicating junctions is the gap junctions. In all those different type of cell junctions, there are different types of cell adhesion molecules present, right? We will see how these different type of cell adhesion molecules makes different type of cell junctions and how the different type of cell junctions are physiologically important for tissue or cells. This is the general functional features of cell junctions. So what is the general functional features? Say this is the first one called tight junction. Tight junctions are junctions which makes contact between adjacent cells very tightly, which makes a seal, right? So tight junction seals the gap between epithelial cells. This is one epithelial cell and this is the adjacent one, right? So it makes the seal between epithelia, right? And due to the presence of this tight junction or due to the presence of that seal, right, it prevents the vertical flow of the molecules from apical regions to the basal surface. Clear? It means if you see the type of transport across the cell, it is categorized into two types. One is called paracellular transport, one is called transcellular transport. So what is the difference? In case of paracellular transport, what we see, the molecules passively can move from apical region to the basolateral surface, right? In passive process based on the concentration gradient of that molecule. And transcellular transport is basically a active transport process where some transporter is required, where energy molecules is required, like transport of glucose by sodium glucose transporter, where molecules always pass through the cell from apical area to the basolateral surface. Clear? But the presence of this tight junction, it forces the transport of important molecules through the transcellular transport process, not the paracellular. If it allows the molecule cross from apical area to the basolateral area by paracellular, it will not maintain the polarity of the cell. So here, the presence of these tight junctions actually helps to maintain the polarity of the cell. So what does it mean? It means due to the presence of that tight junctions, the cells maintains the concentrations of important molecules in both the side according to the need of the cells. It prevents the mixing of unnecessary materials from apical area to the basolateral area. The second one called Adherens junction. So Adherens junctions, here another important thing in tight junction is it is not only makes the contact or seals between cells, at the same time there are some important proteins are involved. I'll explain in details. So in case of Adherens junctions, it connects the actin filament bundles in one cell with that in the next cell. You can see this is the transmembrane protein, right? And this is the actin filament presence in these epithelial cell, and this is actin filament presence in the next one. So Adherens junctions connects the actin filament bundle in one cell with the next. This is another important functions of cell junction. Desmosome, it connects the intermediate filaments. See, this is one intermediate filament presence in one epithelium, another one, this is intermediate filaments. So these junctions basically connects the intermediate filaments of adjacent cell. Here's gap junctions, it allows the passage of small water soluble molecules from cell to cell because it is basically a channel forming junction. It contains a specific channel that allows the molecule. Other than this, what we have seen here, another two important types. One is called hemidesmosome. In case of hemidesmosomes, it makes, makes the connections with the cell matrix, but one part of this is attached to the intermediate filaments. But in case of focal adhesions, another important type, it is attached to the actin filament. So this is a general overview about the functional overview of cell junction. Now see what are the most important type. As I mentioned, the cell junctions involves the different types of transmembrane protein. So if you see the natures of the tight junctions, the most important type of protein presence in tight junctions are Occludin and Claudins. These are tight junction protein, these are transmembrane protein. And due to the presence of this tight junction protein, it prevents the passage of water and water soluble substances, right? That doesn't allow the molecule to pass from cell to cell through these tight junctions. It prevents the transport. Keeps the top and bottom parts of the cell separate, so materials move only through the cell, not between them, right? As I've already mentioned, it blocks the paracellular transport process. Instead, it forces the transcellular transport process. See here, these are the protein. These are the transmembrane protein. There are 4 transmembrane protein. These are called Claudin and Occluding. And this transmembrane protein, as we have seen, it is attached to some cytoskeleton molecule. Mainly, in case of tight junctions, these transmembrane proteins are attached to the actin filaments. But the binding or attachments of these transmembrane proteins with the actin filaments need some adapter molecule or adapter proteins. So here, the adapter proteins are nothing. These are zooprotein called zona occludens, right? If you see the types of zooprotein, there are different types like zo1, zo2, zo3 or zona occludens 1, 2 and 3, right? Based on the cell types, you can see different type of adapter protein or zooprotein. So here, these are the proteins, these are the zooproteins that involves to the attachments of these transmembrane proteins to the cytoskeletal filament, mainly actin filament. Other than this, you can see what is the specificity of both the Claudin and Occluding. Though they are transmembrane protein, but the functions is little bit different. You can see it makes the tight seal between cell, Claudin protein, right? Whereas Occluding keeps the seal strong and stops leaks. So this is the most important features, that preventions of leaking of molecules, right? So it keeps the seal strong and stop leaks. So what are the zooproteins involved with this Claudin? Like Zo1 or Occludins 1, 2 and 3, whereas it is 1. And in both the case, the adapter protein helps the binding of Occludin protein or Claudin protein to the actin filaments of cytoskeleton structures. Another important thing present in these structures is junctional adhesion molecules that helps stick together, and here also it is Zo1 and it is attached to the actin filaments. So these are the zooproteins that present in this particular tight junction structures. I hope this part is clear. So let me brief again about the tight junctions. Tight junctions is a type of junctions which helps in the formations of close contact between cells. It makes a specific, it makes a strong seal between cell. Here, number of transmembrane proteins are present, mainly Claudin and Occludins. Along with this transmembrane protein, some adapter proteins are required for making the seal very strong. These adapter proteins are zooproteins, and that zooproteins allows the formations of strong bond between the microfilaments present in the cell cytoplasms with this Claudin and Occludin proteins. As I've already mentioned about the paracellular transports and transcellular transports, you can see this is very common in epithelial cells. Due to the presence of these tight junctions, it prevents the passage of molecules from apical surface to the paracellular, basolateral. But it forces the transcellular transports. Here you can see, due to the presence of some transporter here, it allows the movements of some important molecules from one side to the other, right, which is transcellular. Now, what is the regulatory mechanisms? How the tight junction functions are regulated? Yes, there are mainly 4 different points that needs to mention about the regulatory process of tight junctions. The first one is called phosphorylations of junctional proteins. So, the phosphorylations of both the Claudin and Occludins can either tighten or loosen the junction. So, because this tight junctions is very dynamic. So, based on the requirement of the cells, it can be loose or tight, right? But here, the losing of these tight junctions or tightens, it depends on whether these junctional proteins are phosphorylated or not. So, phosphorylations of junctional proteins is very important. Another important features, as I've already seen, this transmembrane protein or cell adhesion molecules is calcium dependent. So, here calcium ions are essential for maintaining the stability of this junction, right? So, without stability of that junctions, it would not be possible to maintain the paracellular transport or control the paracellular transport. So, it is very vital to maintain that junction. So, the maintaining of tight junctions is also influenced by the availability of the calcium ion, right? So, when calcium level falls, the junctions become leaky. So, it will promote the paracellular transport, right? During inflammations, different type of cytokines like tumor necrosis factor alpha and interleukin beta can disrupt tight junction and increasing the tissue permeability, right? So, other than this, hormones also played an important role. It changes the permeability of a certain type of tubule, like you can see the formations of urine or reabsorption or absorptions of water. See, like vasopressin can strengthen tight junction, especially in kidney tubules to reduce water loss, right? So, these are the major components that played an important role in the regulation of tight junctions. The first one is phosphorylations of junctional proteins, the availability of calcium ions, then the inflammatory conditions, as well as the role of hormone. Now, if you see a specific example, like the transport of glucose. So, we have already seen that the transport of glucose basically occurs by active process. So, here this tight junctions actually promotes the glucose transport. So these examples actually explains how this tight junctions facilitates the transport of glucose by transcellular pathway instead of paracellular pathway, right? So you can see in intestinal lining, mainly the epithelial cells, we know that epithelial cell surface contains certain types of transporter, and that transporter are very specific. Like here in case of glucose transport, the specific type of transporter present in apical surface is GLUT1, right? But if you see the basolateral surface of the epithelial cells, the transporter presence is GLUT2. It means the GLUT protein or GLUT transporter is very much specific for the transport of glucose. Now, how these tight junctions facilitates this process of transport by using GLUT transporter instead of paracellular. So, you can see here, this is the transporter presence in apical surface, that is sodium glucose transporter, and this is the transporter presence in the basolateral surface. Now, as you can see, this is the tight junction presence in the adjacent cell. So here, the presence of these tight junctions, it allows the molecules of glucose through the transporter instead of this passage, instead of through this space, right? Means simple meaning is it facilitates the transcellular instead of paracellular. The same things you can see in the blood-brain barrier. So it is very vital for the brain cells to get the proper materials for the proper functions. So if there is any leak between the blood-brain barrier, so there is a high chances of the entry of different types of toxic materials to the brain cell that can affect the brain cell. So where tight junctions play an important role. So here tight junctions prevents the entry of unwanted materials by facilitating the transcellular pathway. The same things that I mentioned in the glucose transport. So you can see the role of tight junctions in blood barrier formations, and we also can see the same things in blood cerebrospinal fluid barrier. You can see there are certain types of epithelial cells present in curvate plexus of the brain ventricle, and that epithelial cells or adjacent epithelial cells contain some tight junction, right? And these are the blood vessels which carries a lots of materials, including important and less important molecules, which is not necessary for the brain cell. So here's, but if there is any leaky channel between adjacent cell, there is a high chances of entry of those unnecessary materials toward the brain, right? So, but the presence of this tight junction between epithelial cells, it prevents the entry of unnecessary materials from blood vessels to the brain, right? This is the importance of tight junctions. Now, the second one called adhesive junctions or anchoring junctions. So, like the tight junctions, in adhesive junctions also, there are numbers of proteins are involved. So, what are those proteins? What is their features? Adhesive junctions links adjacent cells to each other, as well as it also links the cells to the extracellular matrix. See here, what are the different types of adhesive junctions? Here it is categorized in two groups, cell to cell contact and cell to cell matrix. So the most important adhesive junctions or anchoring junctions is Adhering junctions and Desmosomes. Both the case, it forms the contact between cell. If you see the protein presence here, in case of Adherens junctions, the protein transmembrane proteins is Cadherin, right? And extracellular ligand. Extracellular ligand means the ligand presence in cell matrix, right? It is Cadherin in neighboring cell. Means the Cadherin molecules which is attached to the cell membrane of neighboring cell. And in the inside the cells, it is attached to the actin filaments. So, what are the adapter protein that are present in this type of Adherens junctions? There are numbers of adapter proteins. The most important one is catenin. You can see there are a group of catenin like alpha catenin and beta catenin. Other than this, vinculin, alpha actinin, plakoglobin, these are different types of adapter protein, and those adapter proteins are vital for making the connection between transmembrane protein or between main protein to the cytoskeletal filament. So in case of Adherens junctions, these are the adapter protein, these are the transmembrane protein, right? But in case of desmosome, the most important type of transmembrane proteins is desmoglein and desmocollin, right? And here also some extracellular ligase presence, as we have seen. Desmosomes also makes a contact between cell. So desmogleins and desmocollins in neighboring cells is the extracellular ligand. And the main difference between Adherens junctions and desmosome is the cytoskeletal filament. So in case of Adherens junctions, the cytoskeletal filament was actin filaments, but here it is intermediate filaments. And the most important type of adapter proteins for this particular desmosome structures is desmolectins and plakoglobin. But if you see the cell-to-cell matrix structures, which are also part of anchoring junctions, focal adhesions, the most important type of adhesion proteins is integrin in focal adhesion. Here's the extracellular, it is basically attached to the extracellular matrix, we have already seen, and it is attached to the extracellular matrix protein, which is fibronectin type. And intercellular cytoskeletal attachments is actin filaments, we have already seen the structures. So what is the adapter proteins? Yes, like lactalin and vinculins, the most important one. Other than this, alpha actinin and laminin is also act as an adapter protein. In case of hemidesmosome, right? So these are the transmembrane proteins like integrin, and it also forms the connections with the extracellular matrix proteins, and it is attached to the intermediate filaments present inside of the cell. Let me show you some structural conformations. See, this is the one structures. Here you can say this one. See, Adherens junctions. As I mentioned, these are the protein presence in first cell called Cadherin. This is the protein presence in second cell, Cadherin. Both attached with each other. And this is the adapter protein called catenin. And that catenin is attached to the cytoskeletal filament. Here it is actin. Here it is also actin. At the same time, if you see the desmosome, this is the integral transmembrane protein, desmoglein and desmocollin, right? Desmoglein and desmocollin is present in the surface of one cell membranes. Here also, this is desmocollin and desmoglein, right? But the main difference here is, instead of actin filaments inside of the cells, it is attached to the intermediate filament. This is what is the difference between this and this. Other than this, you also can see the natures of hemidesmosome. In case of this, you can see the intermediate filaments is there, and the most important adapter proteins is lectin, and this is the transmembrane proteins, mainly the integrin protein. And that integrin proteins is attached to the proteins present in the extracellular surface. Here, either it is laminin or fibronectin. I hope this part is clear. Clear? After finishing my lectures, you have to calm the board. Be prepared for that. Yes. You have to calm the board. You have to explain. Yes. You are getting my point now? So be prepared for that. So what is the functional rule of all those types of adhesive junction? So this is the first one. You can see the most important functional features is it holds cells tightly together. It confers mechanical strength. You people are really getting my points, whatever the things I am discussing? Sure? Okay. You can see what is the exact arrangement of different types of molecules. See the proteins, how they are attached, and how the adapter molecules are important for the attachments with the filaments present in the inside cells. So desmosomes prevents epidermal cells from separating under tensions and cardiac muscle cells from pulling apart during contractions. So it is very vital to maintain the integrity of the cells in contractile muscle cell. So that integrity of those contractile muscle cells is basically maintained by the presence of this type of cell junctions like desmosome. I've already mentioned what is the protein presence in focal adhesions and whether it is attached to the cell or whether it is attached to the cell matrix. You see in broader diagrams, this is the integrin proteins and this is the adapter proteins. These are, you can say, these are different types of adapter proteins, and these adapter proteins binds to this integral or cytoplasmic cytoskeletal filaments, mainly the actin filaments. Here also the most important things I have mentioned about the laminin and fibronectins. You also can see the keratin is also present in the cell matrix structures. Another important part is called the gap junctions. So what is the features of that gap junctions? So we have already seen that gap junctions is also known as the channel forming junctions. Gap junctions is the junctions which allows the molecules from cell to cell through this channel, right? But if you see the structural organizations of the gap junctions, it is basically made up of some important protein called connexon, right? And that connexons is a molecules which is made up of some subunit of connexin. In a single connexon, there is a 6 subunit of connexin, right? That makes a single connexon. So here, the connexon presence in one cells is attached to the connexon presence in the adjacent cells, and they forms a channel. And in the channels, there is a passage that allows the molecules to pass through this, right? You can see the structural features. These are the 6 subunit of connexon, connexin, and they makes a connexon, right? This is the cell membrane of the first cell. This is the cell membrane of the second cell, right? And both the cells there is a single connexon, right? And they forms a channel. See, this is the channel that allows the molecules to pass from the inside of the cell cytoplasm to the cell cytoplasm of that particular cell, right? Now, what are the other important features of these gap junctions? These channels are usually in open state, but will close when the cells become leaky and when the metabolic rate is depressed. This is one conditions when these gap junctions act. It allows the passage of small molecules such as different types of metabolites and inorganic ions to form electrical synapse. So it means it directs the transmissions of action potentials without any transport and its transmitter or without any receptor. The transfer of different types of nutrients along with some waste materials takes place through the gap junctions in avascular tissue such as a lens and cornea of the eye. So these are some important features that is related with the gap junctions. You can see this is the exact arrangements how it forms the connexons by arranging all those connexin molecules. So what is the exact regulatory mechanisms of the gap junctions? Like tight junctions, there is a specific mechanism to regulate the gap junction functions. So again, the calcium ions play an important role. High intracellular calcium ions can close gap junctions to prevent the damage from spreading. So this is very vital to maintain the cellular integrity, to prevent the entry of unwanted materials and exit of important materials is influenced by the calcium ions. pH is also important. Acidic conditions can close the gap junctions. And membrane potentials, significant difference in voltage between cells can open and close the channels. So here potentials or electrical potentials of the cell membrane is also very vital. So which is found in nerve cells, right? Phosphorylations is another important part of these regulatory mechanisms. So phosphorylations of that transmembrane proteins like phosphorylations of connection by particular proteins called kinase, it modifies the gap junctions when needed. Whereas certain types of hormones or neurotransmitters also play an important role to maintain the integrity of the gap junction functions. Like certain chemical signals can increase or decrease the junctional permeability that allows the molecules or it prevents the exit of important molecules. So this is what is the regulatory mechanisms, what is the important part of gap junction function regulations. So these are the some cell types where gap junctions can be found, like skin epithelium, endocrine glands, gastrointestinal epithelium, smooth muscle cells, cardiac muscle cells, the brain cells, as well as glial cells. So these are the most important cell types where the gap junctions can be found. So overall, in today's lectures, what I have discussed, let me summarize again. At the very beginning, I have discussed about what is cell matrix or extracellular matrix, what are the different important protein and non-protein components or structural protein components. After that, I have discussed about cell junctions, different type of cell junctions. Along with this, I have discussed what do you mean by cell adhesion molecule, what are the different types, and how this different type of cell adhesion molecules is important for the formation of a different type of cell junctions. And finally, we have discussed about the regulatory mechanisms of tight junctions and gap junctions. That's all. Thank you.